Good day, this is the Professor, Professor CBMD PhD, and today's Tuesday, November 1st, 2022. It is 1:18 and 6 seconds, AM Eastern Standard Time, and today's research, cancer biomarkers. So let's jump to it here. Transistors help identify cancer cell markers. Date October 31st, 2022. Source, Tokyo Medical and Tensei University. Researchers have found that a system based on ion-sensitive field effect transistors, a tiny electrical circuit that is activated by a change in pH, an enzymatic chemical signal amplification, can accurately detect EGFR of breast cancer cells. And this technology has the potential to be used with multiple markers simultaneously to assess cancer patient status from liquid biopsy samples. And the researchers have revealed a new technique for assessing the cancer-related marker using breast cancer cell lines. Detecting cancer-related markers is a powerful way to determine diagnosis, prognosis, and treatment success. Modern techniques are capable of detecting these markers in patient samples, such as blood and urine, which provides a non-invasive way to monitor and evaluate patients. And by circulating tumor cells, which are cancer cells found in the blood, are one of the main targets used to evaluate cancer patients' blood samples. However, it can be challenging to isolate these cells from the blood, and current approaches do not adequately detect both epithelial cell and mesenchymal cell markers, which are important for determining the stage of the cancer. And to create a system that can quickly and easily detect cancer-related markers on circulating tumor cells and potentially other factors in the blood, the researchers used an apparatus called an ion-sensitive field effect transistor, which is a tiny electrical signal that is activated by a change in pH. They encoded these transistors with breast cancer cells and then added an antibody linked to a chemical reporter that causes a change in pH if the antibody recognizes the cells. They found that the chemical reporter, glucose oxidase, successfully detected the expression of epidermal growth factor receptor, a marker of poor cancer prognosis on circulating tumor cell membranes. And furthermore, the strength of the chemical signal correlated with the amount of epidermal growth factor receptor expressed by the cells. More importantly, these results correspond to the levels of epidermal growth factor receptor detected using other techniques, indicating that the ion-sensitive field effect transistor approach accurately identifies cancer-related marker expression on cells. And these results provide proof of concept that an ion-sensitive field effect transistor-based system can be used to efficiently assess patient cancer status based on liquid biopsy samples. Given that the ion-sensitive field effect transistors can be manufactured to be the size of a single cell and assembled in a vast arrays, this technique has the potential to enable high-throughput analysis of cancer cells at single-cell resolution. In addition, the use of multiple antibodies for the chemical enzyme detection step can enable the simultaneous analysis of multiple cancer-related markers. Good day, this is the Professor, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.